picture this for a second. You're standing in front of this massive, just incredibly complex control panel.
Oh, yeah.
We are talking like spinning gears, strands of DNA unfurling, and this glowing, highly specific key sliding into a lock that, activates this impenetrable shield.
Right.
And the crazy thing is, this isn't science fiction. This is the Major Histocompatibility Complex, or MHC. It's the microscopic control room inside your own body.
And inside the bodies of practically every animal on Earth, yeah.
Exactly. And today, we are doing a deep dive into our sources to decode exactly how it works. Because, well, it basically dictates whether you fight off a disease or totally succumb to it.
Yeah, that visual you just gave, it really gets right to the core of it. Because our goal for this deep dive is to, move completely beyond this vague idea we all toss around.
Like just saying you have a good immune system.
Exactly. When you actually look at the biological mechanics, it is not a simple sliding scale of strong or weak.
Right.
It is a precise microscopic lock and key mechanism. It's a genetic controller that dictates, with absolute authority, how your body or a farm animal's body interacts with the whole invisible world of pathogens.
Okay, so let's unpack this. Because the sources we have today, they completely changed how I picture getting sick.
How so?
Well, I think I'm like most people, right? I usually think of the immune system like a guard dog.
Okay, sure.
Like a virus gets into your bloodstream, the guard dog sees the intruder floating by, barks, and attacks. But reading through this first phase in our sources, the one called entry and fragmentation.
Oh, yeah, the fragmentation phase.
Yeah, I realized I had it entirely wrong because, wait, if a whole virus is just floating around inside a cell, why can't the immune system's T cells just sense it and attack? Why are they basically blind to a whole virus?
It definitely seems counterintuitive, I know. But the thing you have to remember is that the immune system is as dangerous to you as it is to a pathogen.
Oh, right, because it can attack your own body.
Precisely.
Yeah.
So it operates under these incredibly strict rules of engagement.
Right.
It cannot simply react to a whole virus floating by. The pathogen, the antigen, it actually has to be physically captured and processed first.
It has to be dismantled.
Exactly. It has to be broken down.
Okay, so if we use an analogy here, your body is like a highly restricted top secret security facility.
I like that.
And the internal security team, the T cells, they aren't allowed to be triggered just by seeing some guy scaling the outside wall.
Right. They can't just start shooting.
Yeah. Instead, the infected cell itself has to grab the intruder, pull them inside, and basically act like an industrial paper shredder.
Yes.
It physically chops up the intruder's ID badge into these tiny fragments, and then it has to take a piece of that shredded ID badge and stick it into a very specific display case on the outside of the cell wall.
That is a brilliant way to visualize the fragmentation process that highly specific display case you just mentioned.
Yeah.
That is the Major Histocompatibility Complex, the MHC. It's a specialized receptor glycoprotein that literally sits on the cell's surface.
Wait, hold on. Let me pause you right there. Glycoprotein. So we are literally talking about a physical structure made of sugars and proteins sticking out of the cell wall, like a microscopic antenna.
Exactly like an antenna, but an antenna with a very specific groove in it.
Okay.
And its only job is to hold up that shredded fragment of the virus for the immune system to inspect, which, brings us to the foundational rule of this entire system.
MHC restriction.
Yes, MHC restriction.
This is the part that genuinely blew my mind when I was reviewing the diagrams.
It's like wild, right? What's fascinating here is that your immune system's T cells, the specialized forces that actually destroy infected cells, they cannot see the shredded viral fragment on its own.
Right.
And they also cannot see an empty MHC antenna on its own. They are programmed to only recognize the exact specific combination of the two.
Wow.
Yeah, the viral fragment must be perfectly bound inside the groove of the MHC molecule.
Okay, so if the key doesn't fit the lock, like if that shredded piece of the virus doesn't perfectly fit into the groove of your specific genetically determined MHC display case, the alarm never goes off.
The alarm never goes off. The immune system remains completely blind.
That is so crazy.
You could have a deadly virus rapidly multiplying inside your tissue, but if your personal MHC molecules are the wrong shape to bind those specific viral fragments, your T cells just float right past.
They just ignore it.
They essentially say, I don't see anything wrong here, and mount zero defense.
It's honestly chilling to think about. I mean, you have all the weapons, but you literally can't see the target because of a microscopic structural mismatch.
Yeah.
So if everything, our entire survival relies on these specific molecular display cases, how are they actually built? Because the sources point out this deeply conserved genomic architecture.
Right, the evolutionary history.
Yeah, if you trace the evolutionary link, you see vertebrates ranging from cartilaginous fish millions of years ago all the way up to you and me, and we all possess these exact same core MHC genes.
Well, nature found a blueprint that worked in the ancient oceans and just maintained it.
Don't fix what isn't broken, I guess.
Exactly. This core genetic blueprint is heavily conserved in our DNA, and it codes for 3 distinct classes of MHC. You've got class 1, class 2, and class 3.
Okay.
And each plays a totally different role in your cellular security system.
Let's break those down, because looking at the biological structure diagrams, they label class I genes as the universal sentinels, and they code for receptors that catch endogenous antigens.
Right, and endogenous simply means threats originating from inside the cell.
Okay.
So if a virus has already broken into cell and hijacked the machinery, class I is the alarm system that displays the evidence on the outside.
Got it. And the data says these class I receptors are found on almost all nucleated cells, which is basically any cell with a nucleus.
Correct.
Which makes sense for a universal alarm. But I actually have to push back on this whole universal label for a second.
Oh, why is that?
Because I noticed a really bizarre expression quirk in the sources.
Okay, let's hear it.
Skeletal muscle and the myocardium, which is your heart muscle, they show little to no expression of class I molecules.
Ah, yes.
Wait, if class I is supposed to be this vital alarm system that tells the immune system a cell is infected, why are our heart muscles basically opting out of carrying the alarm?
It's a great question. And if we connect this to the bigger picture, it actually highlights a brilliant, albeit terrifying, trade-off in immune biology.
Okay.
Think about what happens when a cell triggers that class I alarm.
The T cells come and kill it.
Right. The immune system's response is absolute. It destroys the infected cell to stop the virus from spreading.
Oh.
Now, your skin cells, or liver cells, they regenerate relatively easily.
Sure.
But your myocardium, heart muscle cells, do not regenerate well at all.
Oh, wow. I didn't even think about that. So if your heart cells expressed high levels of class I alarms, a minor viral infection could cause your own immune system to just destroy massive swaths of your heart tissue.
Precisely. The body is essentially negotiating with itself. It lowers the alarm system on critical, non-regenerating organs to prevent catastrophic autoimmune damage.
That is wild.
It accepts a slightly higher risk of a virus hiding out in the heart muscle because the collateral damage of a full immune assault would be fatal.
That is just terrifyingly logical. It's like saying, I'll let the intruder hide in the server room because blowing up the server room is worse than the intruder.
That's a perfect way to put it.
Incredible. You know, seeing how evolution tweaks these dials really makes you wonder. Oh, and the species comparison chart points out that human red blood cells do not have class I MHC.
Right.
But mouse red blood cells do. It's just wild how nature customizes the exact same blueprint depending on the animal.
It really is. And, while class I guards the inside of standard cells, class 2 has a different job entirely.
These are the specialized presenters.
Exactly. They're designed to catch exogenous antigens.
Exogenous, so from the outside.
Right. Threats from the outside that haven't necessarily infected a cell yet, like floating bacteria.
Yeah.
Because of this, class 2 isn't on every cell. It's located exclusively on specialized antigen-presenting cells.
Right, the heavy hitters. We're talking B cells, dendritic cells, and macrophages.
Yes.
They basically act like border patrol, right? Eating the bacteria, shredding it, and then using class 2 to display it so the rest of the immune system knows what to hunt down.
Exactly. They are the professional scouts. Now, that leaves us with the final architectural piece, which is class 3.
Okay.
And this is where the entire mechanism shifts. Because unlike classes I and II, class 3 genes do not build display cases.
Wait, really?
Yeah, they aren't involved in antigen presentation at all.
Okay, so if class I and II are the security cameras and the alarms spotting the intruders, what on earth is class 3? Are they the ones actually doing the fighting?
Not directly, but they are essential. If we keep your building security analogy, class 3 is the automated indirect defense network, like the sprinkler system and the emergency protocols.
Oh, okay, so what exactly is the sprinkler system spraying? Is it just like generic white blood cells rushing in?
It's a complex chemical arsenal.
Yeah.
Class 3 genes actively manufacture the chemical weapons and support proteins. For instance, they code for the complement system.
Okay, what does that do?
Think of it as water flooding the hallways. These proteins physically coat the pathogens so they can't move, basically marking them for destruction.
Okay, flooding the hulls. The sources also mentioned vital enzymes like 21-hydroxylase A and B.
Right.
And something called tumor necrosis factors, which, I mean, that sounds aggressively lethal.
Oh, they are? They orchestrate inflammation and actively cause targeted cell death to stop tumors and infections from spreading.
Wow.
And finally, Class 3 governs the stress response via heat shock proteins.
Okay, so if the complement system is the water flooding the halls, the heat shock proteins are like the emergency blast doors slamming down to protect the cellular structure from the stress of the attack.
That is a fantastic way to synthesize it. So you have this incredibly elegant, universal blueprint, classes 1, 2, and 3. Sentinels, scouts, and chemical defenders.
Yeah.
But here's where we hit a major pivot point in the biology.
Okay, what's that?
While the 3 classes are universal, the actual genetic arrangement, like the numbers of these genes, the specific shapes of the locks, it becomes incredibly chaotic when you move from species to species.
Yeah, alphabet soup is what I started calling it in my notes. Because to make sense of this genomic variation, scientists created this universal naming system.
Yes, they did.
It generally takes the format of the species name, followed by leukocyte antigen. So for us humans, we have HLA. Dogs have DLA. Rabbits have RLA.
Right.
And in the livestock industry, cattle carry BoLA, horses have ELA, and pigs have SLA.
It is quite the alphabet soup. But, the letters are just the surface. When researchers actually map these genomes, the structural differences between animals are just staggering.
How?
Well, take cattle, the BoLA system. Their MHC complex is massively spread out across 4 distinct regions on chromosome 23.
Wow, okay.
But then look at sheep. Their system, the OLA, operates with just 3 specifically expressed class I genes.
And the numbers just keep swinging wildly from there. Like, the data shows that horses, their ELA system, possess exactly 2 O-class genes.
Just 2?
But then you look at goats, the CLA system, and they are highly multiplexed. A goat carries anywhere from 10 to 13 distinct class I genes.
Which raises an important question for you to think about, doesn't it? What does it mean for daily survival when a goat has up to 13 distinct genetic alarms to present viral fragments, while a horse only has 2?
Well, thinking about what we discussed earlier with MHC restriction, if the shredded virus has to fit perfectly into the display case, my immediate thought is that a goat must be capable of recognizing a much wider variety of viruses right out of the gate. Whereas a horse, with only 2 types of display cases, might be incredibly specialized. I mean, imagine walking through your daily life with an immune system that can only recognize 2 specific architectural shapes of threats. It feels like a massive vulnerability.
It does. And that is the ultimate evolutionary puzzle. The numbers and arrangements differ because they were shaped by the specific pathogens those species historically encountered in their unique environments.
Right.
Which brings us to the absolute core of this deep dive. Because these MHC molecules are the regulators of immune function, these genomic variations, whether you have BoLA or ELA, 2 genes or 13, they are not just trivia.
No, not at all. They are the literal bridge from abstract genetics to physical disease susceptibility.
Yeah.
Because if your specific MHC molecule cannot effectively bind to a pathogen's fragment, the immune system cannot mount a defense. Therefore, possessing certain exact alleles like certain shapes of locks, it dictates whether an animal is highly susceptible to a disease or practically immune to it.
Let's examine how this plays out in the real world. Let's trace the causality in cattle, because this is where theoretical science becomes vital agricultural reality.
Okay, looking at the cattle health data, it is incredibly specific. For example, if a cow possesses a specific allele called BoLA12, the data firmly associates that with a high susceptibility to bovine leukosis, or BLV.
And we know why now. The AI12 display case is fundamentally the wrong shape to catch and display the BLV antigen.
So the key doesn't fit.
Right. The T cells just walk right past the infected cells.
But if that exact same cow possesses the allele BoA07 instead, they show strong, unique resistance to that exact same disease.
That's amazing.
It is a literal binary switch encoded in their DNA. The A07 lock perfectly catches the BLV key, and the immune system destroys it immediately.
Exactly. And it extends far beyond a single virus. The shape of these locks dictates vulnerability across an incredibly wide spectrum of threats.
Yeah, the sources show it dictates susceptibility to parasites like the tick species Buphalus microplus. It affects their defense against squamous cell carcinoma, which is cancer.
Yep.
And here is a massive one for the dairy industry. mastitis defense.
Oh, yeah?
The biological resistance to mastitis is firmly located on the allele BoLA A16. If you have a dairy cow with A16, her cellular display cases are physically optimized to catch the bacteria that causes udder infections. She just naturally fights it off before it even becomes a problem.
And what's fascinating is that we see these direct correlations in every single species we map. Consider the equine health impact data, the ELA complex in horses.
Right. Because the horses only have those two class genes, so the specific alleles they inherit matter immensely.
Exactly.
There's an allergic condition called Kulicoides hypersensitivity, which is basically a severe, debilitating reaction to insect bites. And the sources show this is directly linked to the presence of a specific ELA allele, AW9. If the horse has AW9, their immune system completely misinterprets the insect saliva, and they suffer terribly from the bites. I mean, whether a horse develops sarcoid tumors, which are fibroblastic skin tumors associated with a papillomavirus, literally depends on their ELA makeup.
It's incredible, and perhaps the most vivid, complex illustration of this entire concept is found in avian health.
The chickens.
The chickens.
Yeah.
The data on chicken bee haplotypes provides a perfect sliding scale of how MHC variations dictate survival against Marek's disease, which is a highly contagious viral infection in poultry.
Okay, I want to pause on the word haplotype real quick. We're just talking about a specific group of genes that a chicken inherits together from one parent, right?
Yep.
Like a prepackaged set of genetic locks.
It's spot on. A bundled security system.
Okay, because this avian data is wild. It basically color codes the chicken's survival rates based on which haplotype bundle they inherited. At the far red end of the spectrum, extreme susceptibility. If a chicken carries the B1 haplotype, its locks simply cannot bind the Marek's virus. It experiences a really high adult mortality rate.
And it gets worse. If a chicken carries a combination of B1 and B4, the immune system doesn't just fail to see the virus, it fundamentally misfires.
What do you mean?
The lots get confused and start grabbing the body's own healthy tissue fragments, presenting them as threats.
Oh, no.
Yeah, these birds become highly susceptible to autoimmune thyroiditis. They basically attack themselves.
That is tragic, but biologically fascinating. But then you hit the middle of the spectrum in the data, the B5 haplotype.
Yeah.
You called this out in your notes, and I need you to explain it because the causality here is a huge trade-off.
It is. The B5 haplotype is a phenomenal example of a pathogen response variant.
Go.
Chickens with the B5 bundle are still susceptible to Marek's disease. Their locks aren't great at catching that specific virus. However, their specific MHC switchboard compensates in other ways. They surprisingly show a much better overall antibody response, and their locks are perfectly shaped to catch a completely different pathogen, a parasite called E. tenella.
Oh, wow. So they lose the battle to the virus, but their unique genetic setup makes them practically bulletproof against the parasite.
Exactly. It's an evolutionary compromise.
And then finally, you hit the green zone of the data, high resistance.
Yes.
If a chicken carries the B2, B6, B14, or B21 haplotypes, their display cases perfectly present the Marek's virus. The T cells wipe it out instantly, and they exhibit high vitality.
So if we step back from the raw data for a second, why are we mapping every allele of a cow's chromosome 23 or tracing the exact haplotypes of poultry flocks?
Because it completely changes the future of agriculture. I mean, we aren't just reacting to sick animals anymore. Understanding the mechanics of the MHC isn't just academic trivia.
Not at all. By mapping these specific histocompatibility molecules, we are unlocking proactive veterinary genetics.
Think about what this actually means. By identifying these exact alleles, like hunting down BoLA A16 for mastitis resistance in a herd of dairy cows, or selectively breeding chickens that carry the B21 haplotype, the agricultural industry can actively implement genetic selection.
Exactly.
They can literally breed herds and flocks that are inherently shielded against devastating diseases right down at the DNA level. The shield is built into the blueprint before the animal even takes its first breath.
It represents a total paradigm shift.
Yeah.
We are utilizing the ancient, deeply conserved biological machinery of the immune switchboard to bypass the need for endless chemical treatments and antibiotics.
We're just letting nature do its thing.
Yes, we are simply letting the animal's own precise genetic locks and keys do the work they evolved to do over millions of years.
So let's zoom out and recap this whole journey we've been on.
Okay.
We started with the entry and fragmentation phase, that incredible realization that a tiny shredded piece of a virus has to fit perfectly into a molecular display case called the MHC for the immune system to even realize there's a threat. We traced the ancient evolutionary roots of these universal sentinels and chemical sprinkler systems across the animal kingdom. And we ended up seeing how utilizing those exact genetic codes can predict and literally prevent whether a cow will develop a tumor or a chicken will survive a deadly virus.
It really is a remarkable testament to the hidden precision of biology. But I think there is a broader perspective to mull over here as we wrap up.
Okay, let's hear it.
We've just spent this entire time looking at how tracking exact alleles like BoLA O16 in cattle or ELA-AD9 in horses can literally design a disease-resistant farm animal.
Yeah.
It raises a truly fascinating question about you, the listener.
Oh, here we go.
Think about your own body for a second. If the vitality and disease resistance of all these animals is so strictly and absolutely governed by these invisible biological switchboards, what undiscovered locks and keys are hiding in your own unique genetic code right now? Your specific human leukocyte antigens, your personal HLA setup, is secretly dictating every cold you catch, every allergy you suffer from, and the immunities you naturally possess without ever even knowing it. Your entire health history is, in many ways, just a reflection of which microscopic fragments happen to fit perfectly into your personal genetic display cases.
Now that is a thought that is definitely going to stick with me next time I manage to avoid the office cold while everyone else gets sick. I'll be wondering, was it luck? Or did I just have the perfect molecular display case?
Probably the display case.
Well, thank you so much for joining us on this deep dive. As always, keep exploring, keep questioning the mechanics of the world around you, and we'll catch you on the next one.